Hi, hello. This is Ramesh Kumar Lalkota, Assistant Professor of Botany, MBS Government Arts and Science College Autonomous, Mahabubnagar, Telangana State. Okay, we were discussing about respiration in our last two classes. We discussed about glycolysis. We discussed Krebs cycle. Today, we will see electron transport system in respiration. Before going to electron transport system, let's have a quick look about what we learned yesterday in our last class. Okay, we saw the structure of mitochondria. We saw what is cellular respiration, what how glycolysis takes place, where it takes place, what are the end products of glycolysis. We also saw, mm, see, uh, as a result of glycolysis, two molecules of pyruvic acid formation takes place. Pyruvic acid is a three carbon compound, glucose is a six carbon compound, and in glycolysis, the net gain of ATP are 2 and NADH 2. Okay, so this is the uh, energy, or there is the energy rich compounds NADH and ATP account in glycolysis. Then we saw oxidative decarboxylation of pyruvic acid. Pyruvic acid, as such, doesn't enter into the Krebs cycle. It first undergoes oxidative decarboxylation, and the acetyl coenzyme A is formed. This acetyl coenzyme A is the connecting link between the glycolysis and Krebs cycle. It enters into Krebs cycle. Okay. Immediately after the formation of pyruvic acid, it enters into the mitochondrial matrix, where it undergoes oxidative decarboxylation, and acetyl coenzyme formation takes place. This acetyl coenzyme A enters into the Krebs cycle. Okay, so in this reaction also, one NADH, energy rich compound NADH formation takes place. Okay, so for each molecule of pyruvic acid, one NADH. So for two molecules of pyruvic acid, two NADH formation in oxidative decarboxylation of pyruvic acid. You have to remember all these things. Okay, then we saw about Krebs cycle. It is also known as tricarboxylic acid cycle or citric acid cycle. It takes place in mitochondrial matrix. Acetyl coenzyme enters into the mitochondrial matrix, where uh, it is first accepted by oxaloacetic acid, which is a four carbon compound. Okay, after accepting acetyl coenzyme, it forms citric acid, which is a six carbon compound. Two carbons plus four carbons gives a six carbon compound in the presence of the enzyme called citric synthetase, and coenzyme A is liberated in this reaction. Okay, so these three, citric acid, cisacononic acid, isocitric acid, and oxalosuccinic acid as well, are the tricarboxylic acids. So we call this cycle as tricarboxylic acid cycle, and citric acid is the first formed compound, so it is also known as citric acid cycle. Okay, so in Krebs cycle, we saw that a total of four oxidation reactions takes place. So this is the first oxidation reaction, where NAD acts as an oxidizing agent, and it itself undergoes reduction. And here also alpha ketoglutarate acid, which is a very important intermediary compound, which forms a precursor for many of the amino acids. Okay, so here total four oxidation reactions: one here between isocitric acid and oxalosuccinic acid, one between alpha ketoglutarate acid and succinyl coenzyme A, one between succinic acid and fumaric acid, and the last between malic acid and oxaloacetic acid. A total of four oxidation reactions and two uh, decarboxylation reactions. See here, this is the first decarboxylation reaction, where oxalosuccinic acid undergoes decarboxylation in the presence of oxalosuccinic decarboxylase enzyme. Here, one carbon dioxide is released, and a six carbon compound, one carbon dioxide release, it forms five carbon alpha ketoglutarate acid. Immediate reaction, this alpha ketoglutarate acid again undergoes oxidation as well as decarboxylation reaction. By decarboxylation, it loses one more carbon, and four carbon succinyl coenzyme formation takes place. So, as a result of this Krebs cycle, we have, for each molecule of acetyl coenzyme, we have three NADH, one FADH, and one ATP. So, for two uh, molecules of acetyl coenzyme here. We have a total of 6 NADH, 2 FADH, and 2 ATP. Okay, so all these are energy-rich compounds. These compounds they store energy in the bonds. So these are reduced forms, and during the electron transport system, which occurs on mitochondrial inner membrane. Okay, so there these energy-rich compounds, they undergo oxidation. They give the electrons and protons to oxygen, which is the ultimate acceptor of these electrons and protons. Okay, the protons will be given to the oxygen. It reduces to water molecule, and the energy which is stored inside these reduced compounds is liberated, and it is stored in the form of ATP, adenosine triphosphate. Okay, how it occurs, we will see now. So, electron transport system. We need to know about the multiple enzyme complexes which take part in electron transport system, and different mobile electron carriers, that is ubiquinone, cytochromes, and proton translocation sites, from where to where these protons are translocated, and how many number of protons are translocated, and mode of ATP formation. These are the things which are involved in electron transport system. So we need to know about these things. Okay, so the first one is multiple enzyme complexes. A totally 5 enzyme complexes take part in electron transport system, out of which 1 to 4 enzyme complexes, they purely involve in electron transport, and the fifth one involves in ATP formation. This is the fifth one, is the ATP synthetase. Okay, so we will see one by one these multiple enzyme complexes. uh, the first one, the first complex, we call it as NADH dehydrogenase, or NADH quinone reductase, or ubiquinone oxidoreductase. It is the largest of the 4 enzyme complexes. We have 4 enzyme complexes which help the electron transport, and the fifth one, ATP formation. Okay, so out of the four, this complex one is the uh, largest of all the four. Okay, it contains flavin mononucleotide, FMN, flavin mononucleotide, as cofactor, or you may call it as a prosthetic group also. Okay, cofactor prosthetic group. Okay, it also contains 22 to 24 iron sulfur proteins in 5 to 7 clusters. These 22 to 24 iron sulfur proteins are arranged in 5 to 7 clusters, and this complex helps the electrons, it transfers the electrons from NADH to ubiquinone. This is the first complex, NADH dehydrogenase, or ubiquinone oxidoreductase. It has FMN as cofactor. It has 22 to 24 iron sulfur proteins in 5 to 7 clusters, and this complex helps to transfer electrons from NADH to ubiquinone. Okay. Then complex 2. This is known as succinate dehydrogenase, or succinate quinone reductase. It has flavin adenine dinucleotide as a cofactor or prosthetic group, and it has 7 to 8 iron sulfur proteins. 7 to 8 iron sulfur proteins in 3 clusters, and it also has a cytochrome B560, and this complex transfers the electrons from succinate to ubiquinone. Okay. Then the third complex, this is called cytochrome C reductase, or ubiquinol cytochrome C oxidoreductase complex. It contains cytochrome B and cytochrome C heme proteins. These are heme proteins, and one iron sulfur protein. Okay, and this complex transfers the electrons from iron sulfur protein to cytochrome C via cytochrome C1. That is, the electrons first transfer from iron sulfur protein to cytochrome C1, then from cytochrome C1 to cytochrome C. Okay, so this is about complex 3. And the fourth one, that is the complex 4, cytochrome oxidase. It contains cytochrome A and cytochrome A3 as electron carriers, and oh, sorry, and it transfers electrons from cytochrome C to cytochrome A3 and ultimately to oxygen. Here it is wrongly printed. I'm sorry. Actually, what happens here is this cytochrome C transfers electrons to cytochrome A3, and cytochrome A3 to first of all it is transferred to cytochrome A, from cytochrome A to A3, and ultimately to oxygen. This is wrongly printed here. I'm sorry. Okay, so this is about cytochrome oxidase. Now we will see how this electron transport actually occurs. Okay, the mechanism of electron transport. Okay, you have to observe this diagram. Very easy to understand. Don't get panic by seeing all these things. It's very easy. See, this is the complex 1, enzyme complex 1, NADH dehydrogenase. Okay, this is complex 3, that is cytochrome C reductase, and here this is complex 2, and this is complex 4. All these are enzyme complexes. Where these enzyme complexes present? These are enzymes are nothing but proteins, so they are embedded, they are present embedded in inner membrane, mitochondrial inner membrane. This is the inner membrane. This whole structure is inner membrane, and this is the mitochondrial matrix. This is mitochondrial matrix, and this is inter mitochondrial membrane space. This is interspace between the inner membrane and outer membrane. This is the inner membrane, and here it will be outer membrane, and in between these inner membrane and the outer membrane, this is intermembranal space. You need to understand first. Okay, this is inner membrane. All these reactions are taking place in on the inner membrane, mitochondrial inner membrane. Okay, so in the first step, two electrons are transferred from NADH. See here, NADH to ubiquinone. These electrons are transferred from NADH to ubiquinone. And here the coenzyme FMN acts as a carrier. Okay, so these two electrons first reach FMN. From there, iron sulfur proteins translocate these two electrons to ubiquinone. Okay, uh, ubiquinone is a electron carrier, and this is the ubiquinone cycle, where first it is present in the form of in the oxidized form, that is ubiquinone. Later on, by accepting electrons, it is reduced to ubiquinol. Okay, by accepting electrons, it forms ubiquinone, and ubiquinol, initially it is in the form of ubiquinone, that is oxidized form. In the oxidized form, we call it as ubiquinone. After accepting the electrons and protons from the mitochondrial matrix, it undergoes reduction, and that reduced form is ubiquinol. Okay, ubiquinol, ubiquinol. So this is the first step 1, where do we get this NADH are formed in glycolysis, in oxidative decarboxylation of this pyruvic acid, and in Krebs cycle. These energy rich compounds, now they undergo oxidation in the presence of oxygen. Okay, so here these electrons are liberated, and these electrons, it undergoes oxidation. This NADH, after losing electrons and protons, it forms into NAD plus. Okay, so these electrons are first accepted by this FMN, flavin mononucleotide, which is a coenzyme or prosthetic group. So from this, the electrons are transferred to ubiquinone. This transfer is done by iron sulfur proteins from FMN, that is flavin mononucleotide. This iron sulfur proteins carry the electrons to ubiquinone, where ubiquinone is reduced to ubiquinol. Okay, so this is the first step. Okay, so oxidation and reduction transformations of a ubiquinone play an important role in the electron transport. Then the second step. One more thing here. Um, as the two electrons transfer from this NADH to FMN, four protons from mitochondrial matrix, they enter into the intermembranal space. You can see here, these four protons, they enter from matrix into the intermembranal space. This is the intermembranal space. Via this enzyme complex, these four protons enter into the intermembranal space. This is very important. You need to know about. You come to know about these protons at the end of this topic. Okay, so you need to remember in the first step, while transfer of two electrons, four protons from mitochondrial matrix enter into the intermembranal space. Okay, now the flow of electrons through the complex 3 is linked to Q cycle. See, from complex 1 they are moving to complex 3. So the flow of electrons um, through the complex 3 is linked to ubiquinone cycle. Okay, so it is after accepting the electrons, it is in reduced form, ubiquinol. This reduced form, again it loses electrons, and these electrons are transferred to cytochrome B of complex 3. Cytochrome B of complex 3. So okay, here uh, after reaching cytochrome B6, the electrons ultimately move to cytochrome C1. So from ubiquinone to cytochrome B, and from cytochrome B to cytochrome C1. While the transfer of these electrons in the complex 3, four more protons enter into the intermembranal space. In step 1, complex 1, four protons enter into intermembranal space. In the same way, here also in the complex 3 also, the transfer of electrons between cytochrome B6 to cytochrome C1, the four protons from mitochondrial matrix enter into the intermembranal space. Okay, so now we have electrons with cytochrome C1. This cytochrome C1, from this cytochrome C1, electrons move to cytochrome C. This is also by accepting the electrons, it undergoes reduction, cytochrome C, and by releasing the electrons, it undergoes again oxidation. So this cytochrome C1 transfers the electrons to cytochrome C. From cytochrome C, they enter into complex 4, where these electrons are received by cytochrome A, and this cytochrome A passes these electrons to cytochrome A3, which ultimately move to oxygen and reduce the oxygen to uh, water. Okay, here also you see four protons. Out of four protons for two electrons, only two protons enter into the intermembranal space, whereas two more protons are there. No, these two protons, they move to oxygen to reduce it to water. Electrons come from cytochrome A3, and protons from mitochondrial matrix reach the oxygen, and oxygen is reduced to water molecule. So ultimately the electrons are reached oxygen, and it is reduced to water. Okay, so from A3 to A2, cytochrome A to A3, these copper proteins, they help transfer the electrons from cytochrome A to cytochrome A3. So we have as a whole this complex 1, complex 3, and complex 4 that take part in oxidation of NADH. But we have FADH also, which are formed in Krebs cycle. So these FADH, they won't enter into complex 1, they enter into complex 2. Okay, from complex 2, see here, FAD is a coenzyme. In the same way, the two electrons, they enter into complex 2, and here FAD acts as a coenzyme. This accepts the electron, and from here the electrons move to ubiquinone. Okay, and here 7 to 8 iron sulfur proteins are there in 3 clusters. These iron sulfur proteins help to transfer these electrons to ubiquinone. Again, same thing. From here on, everything is same, but you need to understand one thing here. These 4 protons are not here. When it enters into complex 2, only 4 plus 2 protons, that is 6 protons, enter into the intermembranal space. But when it enters through complex 1, 4 plus 4 plus 4, total of 10 protons enter into the intermembranal space. Okay, so ultimately the electrons, either they may enter from complex 1 or complex 2, they reach the oxygen. Okay, and oxygen is reduced to water by accepting protons from the matrix. Okay, mitochondrial matrix. So this is about electron flow in the inner membrane of mitochondria. See, this is the whole thing. These are the proteins. Enzymes are nothing but proteins. They are found embedded in the inner membrane of the mitochondria. Okay, so for transfer of electrons from NADH, we have 10 protons enter into intermembranal space, and for uh, this FADH, we have only 6 protons that enter into intermembranal space. In cytosolic NADH also, cytosolic NADH, that is the NADH which is formed during glycolysis also, it also enters into complex 2. It also enters through complex 2. It won't enter through complex 1. So those two NADH which are formed during glycolysis also enters through this complex 2, and they also here also only 6 protons enter into the intermembranal space. This is very important. You have to remember the NADH which are formed during glycolysis. They glycolysis takes place in cytoplasm. So the cytosol NADH also enters through the second complex and results in the transfer of 6 protons into the intermembranal space. This is very important. Okay, so this is about your electron transport. Now what happens next? What happens there will be a higher concentration of protons here in the intermembranal space when compared to this mitochondrial matrix. When compared to mitochondrial matrix, there is a high concentration of protons in the intermembranal space. So there exists a proton gradient. The pH decreases and proton gradient develops here. Due to high proton concentration, the pH here in the intermembranal space, it decreases, and what happens, a proton gradient develops and a proton motive force develops here, which pumps these protons into the mitochondrial matrix. Through this membrane they won't enter because it is permeable to only water molecules, and no other ions can pass through this inner membrane. So from where it has to pump, so here you can see here, this is the ATP synthase or fifth complex. ATP synthase or F0 F1 particle, we call it as F0 F1 particle. Here this is F0. F0 is a long stalk which is embedded in the inner membrane, and this is the F1 head-like structure, a round spherical head-like structure which is projected into the mitochondrial matrix. See, this is F1 part of ATP synthase, and this is F0 part of ATP synthesis. This is also known as F0 F1, and it acts as a channel for the flow of protons. Okay, so from here, this through this channel, the protons flow from the intermembranous space into the mitochondrial matrix. Okay, so this was explained by Peter Mitchell as chemiosmotic hypothesis. This is also known as chemiosmotic hypothesis, which was explained by um, Peter Mitchell. Okay. So higher proton concentration outside the inner membrane than in the matrix. So uh, the difference between these proton concentrations across this inner membrane and matrix, it results in the formation of a pH gradient. Okay, so because of the higher proton concentration outside the inner membrane, protons return to the matrix down the proton gradient through coupling factor or ATP synthetase. Coupling factor or ATP synthetase. Okay. Hmm. Then, okay. Hmm. So this is regarding your electron transport system. See, for each three protons, one ATP formation takes place. It is similar to that uh, in a hydropower project. What happens? The water is stored, and this water is allowed to flow down, and this water falls on the wheels, and these wheels tend to rotate, and electricity generation takes place. In the same way, all these protons, they flow from this intermembranous space through this channel, and this flow makes the F1 particle to rotate. Here the head is spherical in shape and it has different components, and the flow of these protons make this to rotate, and the energy generated due to this rotation is used for the phosphorylation. The phosphorylation, that is here it is that ADP accepts inorganic phosphate, and ATP formation takes place. Okay, so this is the ultimately for every three protons, one ATP formation takes place. Total we have 10 protons, so 3 ATP formation for each NADH. Oxidation of each NADH yields 3 ATP, and oxidation of each FADH yields, we have only 6 protons, so each ATP is formed by the flow of 3 protons. So we have 6 protons, so a total of 2 ATP are formed from FADH. Okay, so it will look like this. This is complex 1. This is complex 2, through which FADH enter, and the cytosol NADH also enters through this. And this is complex 3, complex 4, where, see, from cytochrome C to cytochrome A1, from A1 to A3, and ultimately reaches the oxygen, where oxygen is reduced to water by accepting protons from the matrix. Okay, you can see here. See, this is the complex 5 ATP synthase, acts as a channel for the proton flow. So these protons move when three protons flow, one ATP formation takes place. Okay, so you can see here. This is the rotary head, which revolves, which rotates. Okay, by the flow of these protons from this channel, which creates an electric force, so that makes this to rotate, and by using the energy, generated here. ATP formation, ADP takes one inorganic phosphate and ATP formation takes place. Okay, so this is the ATP generation. For each NADPH, 3 ATP, and for FADH, 2 ATP, and cytosol NADH also 2 ATP generation takes. See, in glycolysis, total of 80 ATP is produced, out of which 4 ATP, 2 are utilized in the first two reactions, and net gain is only 2 ATP and 2 NADH. Each NADH, from each NADH, we will get only 2 ATP. So 4 from here, 4 from here, but 2 is used. Okay, so net gain 6. And in oxidative decarboxylation of pyruvic acid, we have for each pyruvic acid we have one NADPH. So for two pyruvic acids, two NADPH, and 2 into 3 equal to 6 ATP. And in Krebs cycle, a total of 24 ATP is formed. 2 ATP directly, and 6 for each acetyl coenzyme A, 3 NADPH. So for two molecules of acetyl coenzyme A, a total of 6 NADPH, 6 into 3 equal to 18. And for each FADH, 2 ATP. So for 2 FADH, 2 into 2 equal to 4 ATP. A total of 20 plus 4, 24. 24 plus 6, 30. 30 plus 6, 36 ATP as net gain by oxidation of one glucose molecule. Okay. This is all for today. Tomorrow we will see anaerobic respiration and respiratory quotients. Okay. Thank you. Thank you very much for listening me patiently. Okay.